students, welcome to Detailed Chronicles by ABHIRAM IAS Academy. So today in this session, let's learn about the genome editing. Now, why this is in news? So basically it is in news because India has unveiled the world's first rice varieties by using the genome editing or gene editing methods. Now, as a question, what is gene editing or what is genome editing? So let's search for the answer in this session. Now, why this is in news? So basically, India has become the world's first country to unveil 2 rice varieties by using the genome editing method. Now, first of all, what are genes? What are genes? Not the genes program which used to come in ETV, but here with respect to body genes, what are they? So basically they are the structural components, right? So a gene is a basic structural component or else a basic unit of the hereditary living organisms. That is, it is passed from one generation to the another generation. That is why we do the, if you want to do the paternity test, we go for the DNA test. No, yes or no? Yes. So basically this gene is a fragment of the DNA. That is it is a segment of DNA. What are DNAs? Deoxyribonucleic acids, right? What it contains? It contains 80 GC components: adenine, guanine, thymine and cytosine. So they're bonded with double bonds, and here they're bonded with the triple bond. Clear? Now, what is the difference between DNA and RNA? Basically, thymine is replaced with the uracil. Clear? So these are the differences, basic differences between deoxyribonucleic acid, that is DNA, and RNA. Now further, RNA is single stranded, but whereas DNA is double stranded. The basic science, right? Now further, what are these genes? So these genes is the fundamental and functional unit of the hereditary. Clear? Next. So this gene is a segment of DNA that contains the instructions for the making protein, which in turn perform a vast array of the functions in the body. Clear? This is the gene. Gene is nothing but the basic and structural unit of the hereditary components, that is basically your DNA. Now, let's see what is the news clearly about. So basically, ICAR marks develops 2 genome edited varieties of the rice. Clear? Who has unveiled this? Basically under the supervision of the ICAR. Clear? What is ICAR? Indian Council for Agriculture Research. Done? So Indian Council of Agriculture Research has developed the world's first 2 genome edited rice varieties by using the CRISPR-Cas technology. Clear? That is CRISPR-Cas technology. Now let's see about this technology in detail in the further session. And the 2 institutions which supported these are Indian Institute of Rice Research, which is in Hyderabad, Indian Institute of Rice Research, that is IIAR, which is located in our Hyderabad, and further Indian Agriculture Research Institute, that is IARI, which is in New Delhi. So basically combination of Indian Institute for Agriculture Research, Indian Institute of Rice Research, and the next one is Indian Agriculture Research Institute, which is in New Delhi. One is in Hyderabad and one is in New Delhi, and these help the ICAR. Clear? Now let's see what is genome editing, and in particular let's talk about the CRISPR-Cas technology. Okay. Now what is this genome editing basically? So genome editing is nothing but modifying or altering or adding or deleting even. What we're deleting? The segment of the DNA or the segment of the gene. So you can see here, we are removing the genome here itself, and maybe we'll be adding one more thing, but it is not from the other DNA component, it is from the within the gene itself. Clear? So we are not adding any, like we are not bringing any foreign DNA here. We are just adding or altering or deleting here itself. Clear? That is your genome editing. So basically, genome editing or gene editing is a precise targeted technique that alters the specific sequence of the DNA. So when you're removing or deleting here, the sequence of the DNA is changed. Clear? So just we are altering the sequence of the DNA within the organism's own genome, but we are not introducing any foreign DNA to this. Clear? When we are introducing the foreign DNA, what is it? That is the gene modification. You are completely modifying the gene. Okay. Next. So as we discussed, it is made up of the CRISPR-Cas technology. What is here? CRISPR. So basically, it stands for clustered regulatory interspaced short palindrome repeats. Short palindrome repeats. Here you can see these are the sequences. You're altering these sequences. Why? To make the gene efficient, or our desired DNA gene we are making. Why? In plants, if we consider for the resistance, high resistance, crop yield, to get the high crop yield, and resistance with specific to drought conditions and flood conditions. Clear? Now, what is this Cas protein? This Cas protein or CRISPR-Cas method is nothing but it is performing like the molecular scissors. Now, what are these molecular scissors? For cutting off your hair, you need scissors. Clear? Now, for cutting off a gene, you need the molecular scissors, and that is the Cas9 protein. Cas9 protein, which is acting as a molecule for cutting your, it is acting like a molecular scissor for cutting your gene. Clear? If you want to cut here, cut this part, you require the Cas9 protein. Clear? Genome editing or gene editing, and next is the method which we are using is clusterly regulated interspaced short palindrome repeats. This is the most effective method for the gene editing. Clear? And Cas9 protein is nothing but it is acting like the molecular scissor, which was previously asked in the UPSC prelims. Okay, they have given Cas9 protein is associated with what? Okay, next there is a concept called site direct nucleases, and there are three types basically. Site directed nucleases, and there are three, and the first one is SDN1, that is it involves simple deletion or mutations of the native genes without inserting any foreign material, making the result similar to the natural mutations. You know the mutations, what are they? If we see any mutation that is naturally occurring changes in the plant or animal body itself. For example, if you see the Simlipal Tiger Reserve, what is the mutation happened with the tigers? So you can see black color tigers. Clear? That is the gene mutation. And this observation is only seen in Simlipal Tiger Reserve. Clear? Now their offsprings are also done, their offspring color is also coming like in only black color. Done? Next. Next, let's see what are the two varieties of rice like ICAR have been developed. So two varieties are Kamala from the Samba Masuri, where the gene edited is GN1A. Why they have edited this? For more grains per plant, okay, for more high crop yield. And next is it matures fast. What happens if it is maturing fast? Less amount of water. It consumes less water. Clear? And next variety is the Pusa DST rice one, where the DST gene is being mutated. Why? For the better resistance in the soil conditions, I mean arid conditions. In the arid conditions. And this is helping farmers in the poor soil areas grow rice productively, and it is also giving high productivity in soil, I mean arid conditions. High productivity in arid conditions. Clear? These two are the varieties, that is Kamala and other is the Pusa DST rice one. Clear? Now next. Let's talk about the gene editing applications. Clear? Or genome editing applications or gene editing applications. First is the high resistivity, high preciseness. That means high precision. You can see it edits the specific genome part. Whichever part you require, that part it edits. And next is no foreign DNA. We are not implementing any other part, any other plant's DNA within the genome itself. We are doing the alterations, or we are adding, or we are deleting as our specific required. Clear? Faster crop improvement. And next is the climate resilience. So we saw now basically in drought conditions also the plants are giving high productivity. So they are becoming tolerable with the specific gene modifications, and it is giving higher yields, and it is very cost effective. So lower input cost and also easy regulation. Now, who does the regulation for this gene editing? Basically, it is done by the Genetic Engineering Appraisal Committee, and it comes under the Ministry of Environment, Forest and Climate Change. Who does the regulations with respect to genome editing or gene modifications? It is the Genetic Engineering Appraisal Committee, which is coming under the Ministry of Environment, Forest and Climate Change. Now, if you are having advantages, we'll also have some disadvantages. What is the basic disadvantage regarding this is the ethical value. What is ethical value here? Ethical concern. So basically, when we are mutating the gene, it is also passing to the next future generations. That is, it is implemented in the germline. That is the future generations passage. The same mutation is passing for the future generations also. Clear? That is the ethical concern. And off-target effects sometimes may, when we are altering the gene, the site may be missed sometimes. So missing of exact site, and it is leading to the undesirable traits or undesirable mutations. Next is regulatory challenges. In some countries, the regulation is very difficult, and it is also leading to the biodiversity risk, where the traditional methods of the seeds are being depleted and some new method of seeds are being introduced into the market. The traditional method seeds, so what happens when the traditional seeds are missed? The taste, the resistance, the resistivity and the desired trait is missing all together. Clear? So biodiversity risk, that is gene edited crops, could reduce the genetic diversity in the crops, making them vulnerable to the diseases and also to the climate. And now lastly, as a value addition, let's discuss the differences between the gene editing and also the genetic modifications. Very simple difference is that in gene editing, there will be no foreign particle, that is no foreign DNA. Outside of the other plant DNAs we will be not using. Within the genome itself, we will be editing. But whereas compared to the genetic modifications, that is, we are modifying the complete gene. That means they are introducing the foreign DNA. That means other plants' DNA or other animals' DNA. Clear? So inserting foreign genes into the organism's DNA. But here, whereas coming to the gene editing, so India has done by using gene editing, not gene modification. Gene modification example is BT cotton. Okay. But here, we are using CRISPR-Cas9 method for the gene modifications to improve the productivity and for climate change, for adopting the climate changes, for adopting the high yield and also disease resistant. Clear? So no foreign DNA in SDN1 and SDN2, but in SDN3, that is site specific nucleases, site direct nucleases, we will include foreign DNAs. Foreign DNA in SDN3, but 1 and 2 will not do. Okay, direct gene addition, deletion or any further removal will do. Here foreign DNA is inserted from the other species in genetic modifications. The regulation is very easy for the gene editing, but for gene genetic modifications, we have to go for number of regulatory procedures. Okay, so here strict regulation is there. Clear? Now what is the public perception? See, ethical concerns, when we see, it is very less for the gene editing. Okay, but for coming to the gene modification, you can face more public opposition due to the insertion of foreign DNA. When this continuous mutations will be done, the conventional trait, conventional gene in them or conventional characters in them will be lost, and this will be passed to the germline. Clear? What is germline? It will be passed to the further generations also, so that the original trait is missing in them. Clear? That is why ethical concerns. And for example, if you see GM mustard, approval is not there for GM mustard, but only approval is given for BT cotton. Why? Because BT cotton we are using for making the cloths or any other some fabric related, or in medical fields we are using. But GM crop, directly we need to eat them. So the public concern here is, maybe you will get the same mutations in our body also. The mutations will affect the body also, and this GM mustard waste, the waste from it will be feed to the even animals. So scientists are telling that even the animals, the possibility of mutations can be seen in animals and in humans also. That is why GM mustard is not approved. Clear? Only BT cotton is approved as the example of genetic modification. Though the research is going for mustard, it is not approved. Clear? So now the example here is CRISPR-Cas9 technology modified plants, but here it is BT cotton, golden rice and other GM, that is genetically modified crops. So this is the difference between gene editing and gene modifications. Clear with this topic? Thank you.